like to introduce the first speaker. It's Professor Sergio Baranzini. He's a distinguished professor of neurology from the University of California, San Francisco, and a member of the graduate program in bioinformatics, which is a major strength of his at the Institute of Human Genetics, the Baker Computational Health Sciences Institute, California Institute for Quantitative Biology, and he holds the Heydrich Friends and Family Endowed Chair in Neurology. He is a scientist with a background in clinical biochemistry and human molecular genetics, and moved to California to study more complex hereditary diseases, focusing on multiple sclerosis. He runs a laboratory which has a multidisciplinary approach, and it is currently involved in the analysis of GWAS, of MS in susceptibility and progression, and molecular studies to identify different stages of the disease and differential responses to treatment. He's the principal investigator of the SPOKE project, a large multidisciplinary bioinformatics approach to gather and integrate and analyse all biomedical data, currently supported by NIH and NSF. He's going to speak to us today on genetic and other risk factors underlying MS and update us to 2022. Sergio, it's my pleasure to introduce you.
Thank you. Thank you very much, Bill, and thank you to the organizers for inviting me to this great talk, great event. I've learned a lot from previous talks already. I think I also need to work on my bio to shorten a little bit. I will need your help. All right, so today what I would like to do, as the title suggests, is I'm going to talk about environmental risks and genetic risks to multiple sclerosis. And I'll start first by mentioning that, as many of you know, MS is a complex disease, and by complex, I mean part genes and part environment. And one of the major environmental factors that has been long suspected, but now really strongly confirmed, is the presence of Epstein-Barr virus in MS patients. And this work by Alberto Ascario that was published earlier this year really proves it, where patients from the military were assessed for the presence of the virus, and almost 100% of the individuals, when they develop MS, were positive for EBV, whereas almost half of the controls only carried the virus. So, of course, the virus is very prevalent, but also developing infectious mononucleosis is a heightened risk for MS. Another environmental factor is vitamin D. Vitamin D plays a role in many aspects of immune maturation and function, both for monocytes and macrophages, dendritic cells, effector and regulatory T cells. And even supplementation with vitamin D has been shown to reduce the hazard of relapses. And even in a clinical trial, it was shown that vitamin D reduced T2 lesions over a period of 12 months. Another environmental effect and lifestyle, and Lars Alfredson will surely talk more about this, is the fact that smoking is a risk factor, and this has been unequivocally demonstrated now. The attributable smoking attributable fraction, as you can see over time, has been diminishing, but this is because people smoke less. But nevertheless, is a significant. It accounts for a significant proportion of the risk. But what I would like to talk more about today is our work on the International MS Microbiome Study, which was designed as a truly international collaboration to address the a question of whether gut bacteria, which respond to many environmental factors, including diet most notably, could in any way impact the risk of multiple sclerosis. And the design of the international microbiome study is that such that we recruit in pairs. So for each patient, we recruit a healthy household control. And the reason for this is that they share a common environment, they share diet, and we've shown that. They share whether there's pets in the house, whether there's babies in the house. All of those factors may affect the composition of gut bacteria. And we know that gut bacteria have an influence on the immune system. And the question is, can we design a sufficiently powered study so that it can serve as the reference for the field? And even if it's just descriptive, can we find associations with bacteria that are living in our guts? And to a certain extent, go from there in terms of interventions and what can we do about it. So this study is designed in 4 phases: a phase for sampling, a phase for multi-omics profiling, a computational analysis, and an experimental arm. So the goal of the study is to recruit 2,000 patients and 2,000 controls, and we're about halfway there. And the work that I'm going to present now, that was published last week in Cell, involves 576 pairs of MS and household controls. The way that patients were recruited in different cities around the world, in North America, Europe, and South America, almost 40% of the patients were untreated with any disease-modifying drug. 60% were treated with some common disease-modifying therapies. And these are some of the results. So when one looks at the diversity of bacteria that inhabit the gastrointestinal tract of MS patients, there's no difference in terms of the diversity between patients and controls. And this is something that is not unexpected. This is more related perhaps to diet than to disease, or to diseases that have the gastrointestinal tract as the main target organ. However, when we look at the beta diversity, that is the global composition of bacteria in cases and in controls, there's a difference. And this is the first time that we were able to show differences in beta diversity between cases and controls, either if we only took the untreated population or if we took the MS population as a whole, there's a difference. And when we looked at the kinds of cofactors or covariables that also affect the composition of the gut microbiota, by far the main factor was recruitment site. So this reinforced our experimental design and recruitment strategy of recruiting pairs, because then we did a statistically paired analysis so that for each case we will compare it to its own control so that we will get rid of these confounding factors. Of course, there were other factors that were also influencing the composition of the microbiota, and we had to include them as covariates in the models that we used. So these are some of the results. Just to highlight some bacteria that were more prevalent among MS cases, and these are the ones that are in red, and some bacteria that were less prevalent among MS. And among the ones that were more prevalent, we still find Akkermansia mucinifera, which is one of the bacteria that were repeatedly found more represented among MS patients. But we're finding many, many more now, and some of them are really interesting in terms of what are the metabolic pathways that they represent. And also there were some bacteria that were less represented, and those tend to be bacteria that produce short-chain fatty acids, which short-chain fatty acids are metabolites that contribute to immunomodulation. So it all fits in terms of the inflammatory potential of the bacteria that we harbored and how they communicate with our immune system over long periods of time in genetically susceptible individuals may trigger MS. So this is some of the pathways that we found, and again, I'm relatively pressed for time, so I'm not going to delve in details into this. What I do want to share with you is an interesting sub-analysis that we did with the disease-modifying therapies. We found that here we divided the therapies into orals, the injectables, and the infusion agents. And you see that there are certain bacteria that associate with the kind of therapy that patients are receiving. And this is interesting because as in other fields, for example, in cancer, has been found that the success of the therapy sometimes depends on the presence or absence of certain gut bacteria, because they metabolize the drug into an active metabolite, or because they stabilize the drug, or because other factors. And in the case of multiple sclerosis, what we found, an interesting finding in this work, was that when we did metabolomic analysis from both feces and serum in a large number of patients, this is a panel of short-chain fatty acids, among which is acetic acid, propionic acid, and butyric acid. So 2, 3, and 4 carbon fatty acids. What we found was that if you look at the ratio between feces and serum of interferon-treated patients, it looks like these short-chain fatty acids have lower concentrations in feces than in serum. And this happened mostly for interferon-treated patients. What we hypothesize is that in interferon-treated patients, there's more active transportation of these short-chain fatty acids from the gut into the circulation. And that will be beneficial because the short-chain fatty acids, again, are associated with immunomodulatory effect. And so to prove that, we tested the expression of 2 messenger RNAs that encode for transporters of short-chain fatty acids from the gut into the circulation. And indeed, these transporters were upregulated. So the expression of the transporter was upregulated in the gut. So again, in interferon-treated patients, these transporters are more active, therefore funneling more short-chain fatty acids into the circulation, and perhaps that's one of the contributing factors for the therapeutic effect of this drug. Of course, there's a lot more to be done, and we're interested in following a lot of the leads that this study has provided. And the last part that I want to highlight is the effect of diet. So for each participant provides a very detailed account of their diet through an extensive food frequency questionnaire. And what we know is that in general, MS patients eat healthier than the controls. But this is probably after the disease has been declared, and as a way to maintain a healthier lifestyle, they eat more fruits, more vegetables, more healthy fats, and so on. And what we're trying to find is, are there bacteria that associate with any particular nutrient that we want to facilitate or eliminate from the gut? And this is a work in progress, but here there's 2 bacteria that are associated with a healthy eating index, meaning the healthy or the target proportion of fruits, vegetables, and refined grains that make up a healthy diet associated with certain bacteria, and this is just 2 of them. The idea here long term will be, can we identify an ecosystem or a group of bacteria that can be provided either probiotically or in any other form, so that they can provide the same therapeutic benefit as if someone were following this type of diet. There's a lot more associations that we found, a lot more than we could analyze in a single study. Of course, this is now the basis for subsequent analysis, going back to the lab, going back to the bench, going into model animals, feeding them certain bacteria and analyzing their immune responses, the response to EIE, and so on. So this is all work in progress through our collaborators in the IMSMS. Here, Amit and UPenn is one of our collaborators. We have 11 sites around the world, and it's extremely exciting and rewarding collaboration. So in the last few minutes, I'm going to switch to genetics, because all of what I spoke about environmental factors, these environmental factors play in a genetically susceptible individual. And what I want to show you today is where are we standing in 2022 with our knowledge about genetics. And I'm going to split into the genetics of susceptibility, so the genetics that predisposes someone to develop the disease, and the genetics of progression. So once the disease is present, what are the chances that genetics play a role into whether this person will be in a wheelchair after 10 years or will be running a marathon after 15 years. So this is a figure that really shows, this was put together by George Evers more than 30 years ago, showing that how the genetic relatedness of individuals relates to the lifetime risk. So for twins, which share 100% of the genome, their lifetime risk, if one twin is affected, for the other is about 35%. And this risk decays in a non-linear fashion as the genetic distance increases, and this is typical for many complex diseases. What this shows is that there is a hypothesis, or there is evidence that genetics plays a role in MS. And way back in 1972, we already knew that the HLA was associated with multiple sclerosis. This was done after a few hundred families were analyzed, and of course the human histocompatibility antigen system was tightly associated with MS, and not surprisingly so, because in the MHC is where we encode all the molecules that can distinguish self from non-self and educate our adaptive immune system, and MS being an autoimmune disease, that made a lot of sense. Now, there went more than 20 years before we could detect a non-HLA association, and that required a lot of more samples. So we went from a few hundred families to 1,000 patients, so almost one order of magnitude above that, to get the statistical power needed to identify associations that were not so strong in terms of their magnitude. So the closer an association is to MS, the stronger the association is. And you can see here that it took longer because these associations, although statistically significant, their magnitude was smaller. The HLA had an odds ratio of 3, meaning inheriting a particular HLA allele, in this case the DRB1-1501, will increase the risk of MS 3 times compared to someone who doesn't have it. And in contrast, inheriting an allele here for the interleukin-7 receptor alpha has an odds ratio of 1.3, meaning inheriting that allele will increase the risk for about 25 or 30%. So, but nevertheless, it was important to continue this quest with more patients, because as more patients were added, you see that more associations were discovered. This is in 2010, this is in 2011, 2013, and 2019. So this is the latest paper of the International MS Genetics Consortium, showing 200 associations in addition to the association found with the HLA. And together, they account for as much risk as the association with the HLA. So it is clear that MS is, the susceptibility to MS is genetically determined. Now, the pathways the genetic pathways that are involved in those signals strongly suggest an immunological flavor. Pathways active in B cells, in T cells, and in monocytes all contribute to the susceptibility, we couldn't find much evidence for pathways involved in central nervous system activity. So it seems like at least the evidence shows us that the genetic contribution to risk comes from genetic variants that affect the immune system. Now, how about the genetics of progression? I mentioned that I will talk about a little bit of both. So progression is exactly once the disease has been manifested, to what extent can we account for common DNA variants to contribute to someone progressing faster or slower? And what, in this case, the IMSGC, what we did was study a population of older individuals with relatively long disease duration, and we were able to compute the EDSS and then convert it to MSSS and to age-related MS severity score, so that we have an outcome measure that is reliable over time. And the study design follow a discovery and replication arms. And this is the largest study ever done in MS progression, with a discovery arm of about 15,000 patients and a replication arm of almost 30,000 patients. And this is in collaboration with the Multiple MS Consortium. Almost 8 million single nucleotide polymorphisms were identified in each of these individuals, and then an association study was performed. First of all, before doing an association study with some new phenotype, one wants to know that there is a heritable phenotype. So the computation of heritability was done, and just we were really excited to see that there was a heritability similar to that that we saw with MS susceptibility, about 13%. Just in comparison, the heritability for COVID-19 is less than 1. So there is signal. Now we wanted to see where is it. The next piece of evidence to share our excitement was that the areas of the genome from which that heritability was coming from was different to that we saw in MS susceptibility. In MS susceptibility, as I mentioned before, most of the signal came from immune-related pathways. So this is blood or immune pathways. In the MS severity or progression heritability, most of the signal came from CNS, tissues and cells. So again, this was really reassuring and exciting. So where is it exactly, and how much? Well, we found one marker that surpassed genome-wide significance. So this is a very, very strict threshold of significance, 10 to the minus 8. So a p-value of 10 to the minus 8 really tells you that if you surpass that, then that is corrected for the multiple hypothesis testing that you're doing throughout the genome. And then there was a second marker that was suggestive, and I'm going to just talk about these two. So the first marker was significant in each of the populations that we studied. These are the different strata, and in the meta-analysis, there's a significant effect with arms. The second locus was also significant in all of the populations that we measured, and the p-value of the replication was significant in both cases. So the combined p-value for both variants exceeds 10 to the minus 8. And the interesting thing is that this is practically what it means is for the lead variant, the 24-week confirmed disability worsening, this is the cumulative probability of worsening for individuals who inherit 2 alleles of the associated variant. So basically, the difference between individuals homozygote for the risk variant and individuals who are not carrying that variant is almost 4 years. So carrying that variant accelerates the time to EDSS-6 or the 20-form confirmed disability worsening by almost 4 years. So we think that this is really significant. And again, this is my last slide showing that both of these variants, the first one and the second one, are expressed in the brain. And they're expressed in cells that we can make a hypothesis that might be involved in the resilience of the brain to inflammation, and whereas cognitive reserve, which was a term that was mentioned earlier, might be involved in the resilience of individuals to progression carrying these variants. So with this, I would like to draw my conclusions, just to highlight that MS is a complex disease, involves the genes and the environment. For the environment, we listed EBV, smoking, vitamin D, and gut bacteria. And then the genes part, of course, for susceptibility, there's more than 200 associations. For progression, only one confirmed so far, maybe more in the future as we increase our N, as you saw, for susceptibility. So with this, I would like to thank my colleagues, Adil Harood and Stephen Saucer, who co-led with me the analysis on behalf of the International MS Genetics Consortium, and also my colleagues from the Microbiome program. Thank you so much for your attention.